For many years, we've all convened at regular intervals to talk about what's new in ALS, and typically have focused on some of the exciting molecular biology that has followed from ALS gene identification and risk factor gene knowledge. But what we've had difficulty locking horns with is how we finally understand and begin to treat sporadic ALS. So we thought it would be pretty compelling this afternoon to have several of the leaders who think about this join us to share their perspectives. And I'm just delighted that we have a remarkable lineup, beginning with Professor Ammar Al-Chalabi, who is Professor of Neurology and Complex Disease Genetics at King's College. He is co-director of the National UK M&D Research Institute, King's Care and Research Center Director, and he chairs the International Symposium on ALS M&D, which convenes in December for its annual meeting. He will speak today on the topic of how we understand multi-step events that may lead to sporadic ALS. Ammar, thank you for joining us.
Thank you very much for inviting me, Bob. So this should share my full screen.
Perfect.
Thank you. So I'm going to be talking, as Bob said, about the multi-step hypothesis of ALS. So we know there are two obvious causes of ALS. The first is family history. If you have a family history of ALS, your risk is greatly increased. And the second is age. We know that the older somebody is, the more likely they are to develop ALS. A family history is present in about 5 to 10% of people, and that tells us that genes must contribute to risk. The risk increases with age, and that tells us that something about aging must be related to the mechanism. So if we just explore those in a little bit more detail, genes appear to be important regardless of family history. So we know if you look at people with apparently sporadic ALS, so with no family history, work we've done, work that Dame Pamela Shaw has done, show that about 20% of people have a large effect Mendelian disease gene, regardless of whether they have a family history of ALS or not. And we also know that small effect variants that just nudge your risk towards getting ALS also contribute, and that the risk from those kinds of variants is probably about 60%. We know that from twin studies. Now, if you put all those numbers together, you can show that the burden of genetic ALS is actually greater in the apparently sporadic population than in the familial population. About 80% of the 10% that have a family history will be genetic, whereas 20% of the 90% without a family history will be genetic. And so you've got about 18% of people with apparently sporadic ALS versus 8% of people with apparently familial ALS contributing the genetic pool. And if we explore age a bit more, we can see that the risk of ALS increases with age. So there's virtually no risk really below the age of 40. There's no risk really below the age of 40. And then you can see the steepest part of the curve is around 60 to 70. So that tells us that's the peak age of onset. But you can also see that the risk just keeps increasing as people age. The 1 in 300 risk line is there in red. So 1 in 300 people will develop ALS. You can see the risk is always higher for men than for women. So one way to model these things is to say that everybody is born with a certain genetic load, that there is some cell damage with time perhaps, and that there are environmental exposures. So if we call those G, T, and E, and then if you reach a particular threshold of that burden, then you'll develop a self-perpetuating process that leads to the person dying. So that's one way we can think about ALS. So then we have to explore what are these genetic, time, and environmental components, and how can we find them? So environmental risk factors are actually very difficult to find because the environmental exposure, the envirome, if you like, is infinite. The genome is big, but it's a tractable problem. It's 3 billion base pairs. If you can sequence it in enough people, you can do the statistics and find out which genetic variants might be important. But really getting the same information on environment is very, very difficult because you're relying largely on recall, and it's unclear what questions you should be asking. It might be something as something like what temperature do you have your shower at versus whether your pillow presses your neck versus if your exposed to exercise, etc. It could be almost anything. So knowing which questions to ask becomes very difficult. There have been some environmental risk factors that have been studied in detail. Smoking, military service, pesticide exposure, heavy metal exposure, and exercise have all had at least some replication and have some evidence that they may be important. But that evidence is variable. So some studies have been done showing that they're not a risk factor at all. So it's difficult to replicate these environmental risk factors. And then if you think about ALS in general, you come up with some mysteries. So we know that ALS genes can cause other diseases. For example, the most frequent genetic cause of ALS is expansion in the C9orf72 gene. And if you expand that gene, about 10% of people have that genetic expansion if they have ALS. So it greatly increases your risk of ALS. Only about 3 in 1,000 people in the general population carry that expansion. That genetic variation also increases your risk of frontotemporal dementia. So that ALS gene not only causes ALS, but it also increases the risk of a different condition. And we also know that you can carry that gene variation or others and never develop ALS. So for some reason, not all gene carriers will develop the condition. We know, I've already explained, environmental causes are difficult to find. And we know that the risk increases with age, but why? And why does there not seem to be a single cause for ALS? And why is there this age-dependent gene penetrance that even if you carry a genetic cause for your ALS, you've had that since conception, and yet you don't develop ALS until you're 50 or 60 in many cases? So these properties are very similar to the situation in cancer as well. And because of that, we took a clue from the cancer world where they've looked at a thing called the multi-step model. So the multi-step model posits that if there is a factor that increases your risk of the condition, that will have a particular relationship by age. So your risk of being exposed to that factor will increase as you age. And if you need more than one factor, then you end up, if you look at the maths, it's fairly simple to explain, but I didn't want to put a lot of maths into this talk. You end up with a power relationship between the incidence and the age of onset. And that means if you plot the log of the incidence against the log of the age of onset, it should be a straight line if there is a multi-step process involved. If ALS has, first of all, one exposure, then another exposure in steps. And if you look at the maths, the gradient of that line tells you something about the number of steps. Now, although I talked then about exposures being the factors, they don't have to be. It can be molecular steps. So it could be that one exposure gives you 6 molecular steps. But the important thing is that those steps have to increase in their probability or change in their probability with age. Now, we looked at this in the Southeast ALS Register in the UK. And this is the plot of the log of incidence against the log of age. This was back in 2014. And you can see the R squared for that line is pretty good. It's 0.95. So it fits very nicely with a straight line. And then we went to colleagues in the Netherlands, and in their register, the R squared was 0.99. It fits very closely with a straight line. In Italy, 0.95. In Ireland, 0.99. And in Scotland, 0.97. So all of these different population registers have a power relationship between the age of onset and the incidence of ALS. And overall, the R squared is 0.99. And what that tells you is that all of them had the same gradient. So if we then look at the gradient, it's always around 5. And a gradient of 5 tells you that there are 6 steps, just the way the maths works. The gradient is 1 less than the number of steps involved. And understanding that ALS is a multi-step process that requires, on average, 6 steps to happen explains many of these mysteries. It means that we can explain why ALS genes also cause other diseases. Because, for example, if you need steps 1, 2, 3, 4, 5, 6 to develop ALS, but 1, 2, 3, 4, 5, 7 to develop FTD, for example, then that would explain why the same genetic factor, which is triggering more than or 1 or 2 of the steps, doesn't trigger all of the steps. And so then the other variations, the other exposures will change the disease that's happening. The reason not all gene carriers will develop ALS is because you need to experience all 6 steps for the disease to start. Environmental causes would be difficult to find because different factors could trigger the same molecular step. So, for example, smoking increases the rate of mutation, so does radiation exposure. The increasing risk with age is because it takes time to accumulate all the steps. The lack of a single cause is because, as I said, different factors could trigger the same molecular step. And the age-dependent gene penetrance would be because even though you're born with that gene, it still takes time to trigger all of the different steps. So this model, it's quite attractive as a model. Our colleagues in Australia have replicated it twice. It's also been replicated in a Danish register, in a South Korean register, although interestingly, in theirs, it was only a 5-step model. So that suggests they have a higher risk of ALS because they only need 5 steps to develop the condition. In a Japanese register, it also had a gradient of 6, a gradient suggesting 6 steps, sorry. And in an Italian register, it's been replicated twice as well. So it seems to be a fairly consistent thing that the incidence of ALS obeys this power relationship with the age of onset, and therefore that it's consistent with a multi-step model. Now, you can use this model to try and find out what's going on to some extent. So, and remember, this is in people with apparently sporadic ALS, so it's just in the general population. So if you take from that population the people with a genetic basis for their condition, then you can look at the gradient in that subset of people, and that tells you how many steps the model requires, and therefore how many steps are accounted for by that gene. So if you do that for C9orf72 expansion, which is, as I said, the commonest cause of ALS, at least in European populations, you can show it accounts for 3 of the steps. It only leaves a 3-step model to account for. So 3 of the steps are accounted for by that variation, and then just a further 3 are needed. If you have exposure to the TARD-BP genetic variant, so that's in the gene coding for TDP43, that accounts for 2 steps, and you still need a further 4. And then SOD1 variation accounts for 4 steps, and you only need 2 more before you develop ALS, which would explain why SOD1 variants often have a very high penetrance. You can also use the model to look for environmental risk factors. So remember, the gradient for ALS is 6 steps. This is an Australian study. Naomi Ray, who's now at Oxford, led this study. So if you look at ALS with heart disease, then the gradient shows it's 7 steps, which suggests that heart disease might be protective for ALS, or at least heart disease risk factors. But remember that people with heart disease are at risk in the same way as people with ALS. So the age of onset of heart disease, the age of risk is also in the 50s and 60s. It's commoner in men, etc. So it may well be that there are competing causes of death, and that's why it appears that having heart disease is protective for ALS, because all the people with heart disease who would die have died, and therefore you're left with people who are protected to some extent. Now, if you account for the causes of death, you can show that actually it's a 3-step process in people with heart disease. And that suggests that heart disease actually is a risk factor for ALS. The factors that increase your risk of heart disease increase your risk of ALS. But this competing cause of death interferes with how we would see that epidemiologically. Now, why is heart disease associated with ALS, the risk factors for heart disease? You can try and answer that using genetic studies, again, from people with apparently sporadic ALS. And one method you can use is a technique called Mendelian randomization. And that's a technique where you assume that genetic variations behave a bit like randomization in a clinical trial. So imagine if you wanted to know, does smoking cause lung cancer, but you had no idea whether it did. One way you could carry out an unethical experiment would be to force half the population to smoke from a very early age and completely refuse the other half of the population to ever have access to cigarettes. And then you would simply see if the group that you'd exposed cigarette smoke to from an early age had cancer. And that would show a causative relationship between the two. Now, clearly that's an unethical experiment, but nature does that to some extent because it gives random assignment of genetic variation to us from our parents. And some of those genetic variants will predispose people, for example, to smoke. We know that variants in one of the acetylcholine genes predisposes people to want to smoke. So you can have a proxy for environmental exposure from your genetic variants, and that's called a genetic instrument. And if you look at the instruments for cholesterol in genome-wide association studies, you can show that if you ask the question, is the causal direction from cholesterol towards ALS? So are the variants that predispose you to high cholesterol leading to ALS, are they more frequent in people with ALS? You can show that it doesn't matter what the Mendelian randomization technique you use. That's what these funny words are on the left. They're all different methods of Mendelian randomization. In general, they all show that cholesterol predicts ALS. And if you ask the question the other way around, is it just that ALS happens to increase your cholesterol, perhaps because your diet has changed or something like that, you can show that reverse causation isn't there. So it looks like cholesterol is important as a risk factor for ALS. And actually, you can ask the question another way. You can look at the epigenome, and the epigenome is methylation of the DNA, and that's a way of switching on or switching off genes. And if you look at the epigenome, genome-wide, then again, you can show that cholesterol, which is this light blue cluster, is very overrepresented and is one of the risk factors for ALS. Now, the other group you can see here, there's obviously two other groups, but the other one I want to just concentrate on for a moment is the inflammatory subgroup that comes up again from this genetic study of people with apparently sporadic ALS. So that inflammatory group I just want you to bear it in mind. A study that we did, but has also been done by others in a similar way, looked at the motor cortex, the occipital cortex, and the cerebellum in a set of postmortem brains, and looked at the transcriptome in those brains. So that's the RNA, the different gene expressions for that set of cells. And we could, using a form of deep learning, a form of artificial intelligence, if you like, combined with statistics, show that there were 3 clusters of ALS. And that was consistently found, regardless of how we did it. And actually, we also could show it in 2 different blood sets. Now, the bottom left one there, the D block, is showing that we can also distinguish controls from the cases. Excuse me, it's very dry air here. And we found that there were 3 different ALS subtypes corresponding to synaptic signaling, oxidative stress, and neuroinflammation. So again, there was inflammation there. And we know that in ALS, there is macrophage and microglia activation. And we also know that T regulatory lymphocytes, which are these cells that are like the policemen of the immune system, can predict your progression. Lower levels predict faster disease. And a recent study, a clinical trial showing treatment with low-dose interleukin-2, which increases your T regulatory lymphocytes number and function, that also seems to show that it improves survival and function. So that paper is currently in review. So we know that multiple steps might include the immune genotype, the inflammatory trigger, and an inflammatory response. And retroviruses, which are part of the human genome, would account for that potentially. About 59% of people with ALS have a retrovirus signature in the blood, whereas only 5% of the general population does. And that suggests that this is an endogenous retrovirus, not infective retrovirus. When you look at the relatives and spouses, it's the pattern of inherited disease, not infective. And one of these endogenous retroviruses that we all carry is called HERV-K. And that's activated in some cancers, and it also appears to be activated in ALS. And actually, the protein for HERV-K is toxic to motor neurons, as shown by Avi Nath at NIH. And as a result of that, we've got a phase 3 clinical trial that's designed to act against these endogenous retroviruses. There's also an MND-like, an ALS-like disease in mice, where they have an endogenous retrovirus, and some mice happen to have a genetic variant that means that they express a lot of these endogenous retroviruses. And if they're exposed then again to another virus, murine leukemia virus, they will develop an ALS-like condition. So that's a multi-step model with genetics and an environmental risk factor and endogenous retroviruses. So we can take all of this and go back to our gene time environment model and say, well, look, the 6 steps account for this gene time environment component. And once you reach the 6th step, that's when you get onset. And we know if you have lots of small risk factors that nudge you versus one big Mendelian risk factor, then you might get a different number of steps accounted for, but it's still the same kind of idea. And then if you take that to its logical conclusion, it means that a good way of looking for environmental risk factors would be in people with a very large genetic component, would be in the people with the Mendelian disease genes, because so many of their steps have already been accounted for. So counterintuitively, we should look for environmental risk factors in people with genetic ALS. So in conclusion, genetic and environmental risk factors contribute to ALS, possibly through a multi-step process. The themes appear to be possibly through transcriptomics, through cholesterol metabolism, and through inflammation. These are the sponsors of all the research that you've seen, and these are my team who've done all the hard work. Thank you.
Thank you, Ammar. That's just terrific, as always. I encourage individuals to either raise their hands, which I may not see, or put their questions in the chat. So, do you have any preliminary readout on the anti-HERV-VK therapy? I know that has been tried in other settings, and I'd love to get your take on the initial results, and we can come back to more about that later if time allows.
Yeah, so we don't have any proper results yet. An interim analysis has been done for futility, because that was the trial design. And the trial is currently continuing.
Fantastic.
That's the only readout we have.
And if you don't mind, a coupled question is this: when you say a blood-based retrovirus signal, what does that mean? Is that evidence of actual HERV-VK protein transcript? Can you comment on that?
No, so that was a study where we showed, actually with you, Bob, and with Merit Cudkowicz, that reverse transcriptase was the signal that we detected in blood. So we don't know where that reverse transcriptase comes from, but the HERV-K genome that we all carry does include a reverse transcriptase component.
Excellent.
There are some questions in the chat now, Bob.
Great. Let's turn to those. Yeah. So let me go to the first one, then I'll try to read these in sequence. Melanie Leitner asks, given the potential relationship between ALS and cholesterol, is there an ApoE association with ALS?
So people have looked for an ApoE association with ALS, but none has been found. And my very first paper ever actually was an association of ApoE4 with bulbar onset ALS, but the p-value wouldn't be accepted for a genetic study now. It was something like 0.04. But that got into the Lancet, surprisingly, back in 1994.
Thank you. And Dr. Hornstein, Aaron Hornstein, says, I wonder about the relevance of transcriptomics data from Molly Hamill and the New York Genome Center that have the same main disease subgroups. Are you familiar with that work?
Yes, yes. It is very interesting. So it was completely independently done and comes up with exactly the same 3 subtypes. And I think the important thing about that is what we don't know is, do people have only one of those subtypes throughout their disease course? Does everybody have all 3 subtypes, but perhaps one is dominant? Or do people start in one and then move to another and move to another as the disease progresses? And that's a very important thing to know, because it would affect how you might treat somebody. Should we expose everybody to all, if for example, if inflammatory phenotype responds best to inflammatory treatments, then should we target those people with it? And if it depends on your disease stage, then we'd have to monitor people and then give them that treatment, etc. So it's an important question to answer.
Thank you. Thank you. From Ben Brooks comes this question: is the TDP43 pathology the same in HERV-K ALS?
Yes. So HERV-K ALS is just general ALS.
Thank you. And can you comment, please, a question from Dr. Bhattacharya about the reason that there might only be 5 steps in the South Korean population?
Yes, it is intriguing. So assuming that the study was done correctly, and there's no reason to doubt that because it was the same group that has done all the other studies, it does suggest that there's something in the South Korean population increasing risk. So it might be a lifestyle exposure or some other exposure that a large proportion of the population has.
Thank you. And I think we can conclude questions with one more key question from Dr. Ostrow. And I'll read it. The normal function of every cell in our body involves multiple complex and interesting processes. The biology is stochastic. What sort of progressive disease would not be modeled as multi-step in that context? Can you comment further on that? And,
yes.
Very broad processes that are important in virtually every cell in the body. So
yes,
there's a bit of a generality here that begs some specification.
Yeah, so that's a very important and interesting observation. when we've published our first multi-step hypothesis paper, we modeled another disease that wasn't multi-step, at least in our hands, which was multiple sclerosis. So when we took multiple sclerosis population data, it didn't follow a straight line. It follows a strange curve when you did a log of incidence against log of age. It doesn't mean it's not multi-step. It could be lots of different multi-steps. And when you add them together, they're just not multiple. But I suppose what it shows in ALS is that it's a relatively homogeneous process, whatever's happening.
Thank you. And I'm going to just squeeze in two more quick questions, please. So one of them is that we heard from the CDC presentation from Jamie Raymond that exercise is a potential risk. And I guess...
אני
Hello?
תכף מתחיל לדבר, אז...
Hello?
Aaron, you're not muted.
I can't... Did you want to say something, Aaron? I'm not hearing you.
Yeah, it was a technical mistake, sorry. I'm waiting for my in line to talk later.
Okay, thank you. So briefly, the question is intense exercise a risk? And would that be particularly important in cases that are genetically endowed?
So Pam's work, and Pam has her hand up, would suggest that people with C9 genetic variation are predisposed to ALS if they exercise.
Yeah.
So that particular pathway would be it.
Thank you.
So what we've found, Bob, is if you're a C9 expansion carrier, the higher the level of strenuous physical activity in your adult life, the earlier the disease kicks in. So the earlier the age of disease onset. So I like Ammar's suggestion that to really get a handle on the environmental steps, we should be looking at the genetic subtypes of disease.
That seems very, very important. And if time allows later, I'd like to share an anecdote related to that comment. Thank you. That's very helpful. I can't resist one final question from Karen Sachs. Will the transcriptome signature essentially be evident in induced motor neurons? In other words, are there degrees to which you can correlate the substrates, and subsets in iPSC-derived motor neurons with those in patients?
That's a fascinating question. I don't know the answer. So you take somebody who has an immune in the immune cluster with their stem cell derived. Yes, I don't know.
Thank you.
Interesting thing to do.
Thank you. Wonderful presentation.